Good morning, everybody. So today I'm going to talk about surgical target selection for the subcallosal cingulate region, deep brain stimulation based on the structural connectivity. My name is Sueng Choi. I came from the Emory University. So I have nothing to disclose. So this is brief outline about the today's talk. So I'm going to talk about a little bit background about the subcallosal cingulate deep brain stimulation. And also to do the tractography, we do the modeling for the volume of tissue activated. And the first goal for our research is defining necessary pathway for the effective SCC DBS. And second goal is prospective surgical target selection for the SCC DBS using the necessary pathway, and summary and conclusion. Here is the figure show that how we do the deep brain stimulation operate in the OR. Like deep brain stimulation is an invasive technique. We put the electronode here inside the deep brain, and that's connect to the called the IPG device as a pulse generator and the battery. So we put the IPG device here on the chest. So deep brain stimulation of the subcallosal cingulate provides sustained antidepressant effects in the treatment-resistant depression, we call TRD. So why subcallosal cingulate, or sometimes we call subgenular cingulate or CG25? So when we induce the depressed mood, we can see the high activity at the CG25 region in various studies. Also, when we have a successful treatment with the depression patient, you can see the decreased activity at the CG25. However, it's not only CG25. CG25 is connected to the several brain regions and build a network. So our hypothesis is that TRD is a state of the dysregulated SCC25 connectivity. So here is a brief summary about the SCC DBS. So SCC DBS is a targeted neuromodulation at the critical load I showed you before. So anatomical location of SCC region is here, right end of the under the corpus callosum and end of cingulum bundle here. And we do the SCC DBS is electrical pulse stimulation on the structurally connected brain region through the white matter bundle. As you see on the right figure here, if we stimulate at the contact 3, probably they have a more stimulation to the frontal, like a medial frontal 10. If go to the 4, probably more cingulum. And for the SCC DBS, we do the bilateral stimulation, and each, as you see here, we put the bilateral electrode here, and each electrode have a 4 different contact, as you see here, like a blue dot is each contact we acquired from the CT image. So previous study with health control, we identified structural connection from the small SCC region here. We put the seed here and do the tractography, and we found that that SCC region is connected to the various brain regions like medial frontal and cingulate and the rocks of the subcortical region and also amygdala. So SCC DBS likely operates through the modulation of activity within a network of the regions structurally connected to the stimulation target. Currently, target selection is based on the SCC anatomy, as I showed you before. Like, okay, we look at where is the ACPC line and how far from ACPC line, and look at the T1 image and where we have to put the electrode. However, if we understand what is the necessary pathway for the effective SCC DBS, we might improve clinical outcome with prospective surgical target selection using a patient-specific individual structural connectivity pattern. So our hypothesis, our purpose is the first, defining necessary pathway for effective SCC DBS, and second, prospective surgical target selection based on the necessary pathway to improve the clinical outcome. To do that, we first have to model the volume of tissue activated. So how we do that, probably many people know that is based on the stimulation parameter, we do the FEM modeling thing and for instance, just one example is if you gave them 1 volt stimulation, probably you have this dark gray volume is activated. If you have like a 5 volt stimulation, probably you can get this light gray big area can stimulate. With that, also, as I told you, we have 4 different contacts in each electrode. When we use a C3 contact for the stimulation, as you see, the structure connectivity pattern looks like this. However, when we use the C1 contact, the bottom contact, you're going to see the different connectivity pattern is shown. So based on which contact we have to use, and based on the modeling, we do the tractography to find the necessary pathway. We use the 17 DBS patient, and we exclude 1 patient because of the image quality. And basically, when you see here, after surgery, we take a CT image and identify where is contact location. And then we do the volume of tissue activated modeling using the Cicilline software that's created by the Dr. Cameron McIntyre at the Case Western. And we estimate the volume of tissue activated like this size. Briefly, it's around 120 cubic millimeter based on the stimulation parameter, but it's changed based on 6 milliamp or 3 volt, 4 volt, something like that. And then using this as a seed point, we do the whole brain tractography, probability tractography. And then at the 6 months of the chronic stimulation, we define that the patient, their Hamilton score is decreased to 50% as a responder. If less than 50%, we call the non-responder. First thing, we check their anatomical location of the contact stimulation. Chronic stimulation contact is different between the responder and non-responder. Actually, they are statistically no significant show up between the responder and non-responder. So anatomical location cannot predict the clinical outcome. However, when we look at their connectivity pattern between the responder and non-responder, as you see the 2 figure here, their connectivity pattern looks different. So responder hit all the medial frontal and the ventral striatum and the subcortical region, and also cingulum bundle. However, non-responder stay in local, their connectivity pattern. Also, not only 6 months, when we look at the 2-year outcome, so some subject was a responder at the 6 months, and some subject doesn't. And non-responder, we change the contact or their stimulation parameter, and they become a responder. So totally at 2 years, we have a 12 responder. And when we look at their connectivity pattern at the 2 years, they exactly identical to the 6 months connectivity pattern. So what we found that responders share involvement of a set of pathway intersecting at the STC region. That's the forceps minor bundle, unsynaptic vesiculus bundle, cingulum, and also medial frontal to the stridor or subcortical fiber bundle. Also, when we look at I already told you that some of subject was a non-responder at the 6 months. When we check their responder contact with the tractography at 2 years, you can see the green is their 6 months non-responder tractography pattern, and the blue is their 2-year connectivity pattern. As you see, they get finally with the contact change and stimulation parameter change, finally they get the medial frontal and more to the subcortical region. So with corrected stimulation, contact and parameter, non-responder become a responder eventually. With that kind of all the connectivity pattern, we build a model that this is a necessary pathway for the effective SCC DBS. As you see here, red is forceps minor bundle that pass through the 25 region, and blue is the unsynaptic vesiculus that pass also through the 25, and the cingulum bundle as a yellow, and from frontal to the subcortical to the ventral striatum. So necessary pathway is this is repeat, but forceps minor, unsynaptic vesiculus, cingulum, and the medial frontal to the subcortical fiber. Based on that, we can do the prospective target selection using deterministic tractography. This is one illustration. So with the standard setting, like 130 hertz and 6 milliamp, we can model that this green ball and do the prospectively the deterministic tractography, this illustration showed if we stimulate here, probably we only catch unseen fasciculus. And if we little bit go up, probably perhaps minor unseen fasciculus, but we're going to miss cingulum. Go up a little bit more, probably we can catch the ideal or the bundle. And too much, probably we miss some bundle. So based on necessary pathway, clinical outcome could be improved using patient-specific prospective surgical target selection. So we did that for the 11 subjects. Before we have a surgery, we do the deterministic tractography to find the optimal location as here. And then we gave to the neurosurgeon that coordinate, and they possibly they can have an error, but they put the exact same location as the here. And as you see our template and what we get from the deterministic tractography, that pretty much match well. So using deterministic tractography, we could prospectively find the optimal target location in each subject. Yes. So here is the outcome. Previously, anatomy-based targeting, we have a clinical outcome as a 41%. With the connectivity-based targeting, we improved that to the 72%, like a 31% improved. Still, this small sample is small, so on the power for the statistical significance, but we improved 30% of the clinical efficacy for the SCC DBS. And then also when we look at the second cohort, like 8 subjects from the out of 11, their connectivity probability connectivity pattern showed nearly identical, very similar to the previously what we found. So conclusion is using probability tractography, we found the same connectivity pattern and we improved the clinical outcome 31%. So like a repeat, but we defined the necessary pathway that's perhaps minor unseen fasciculus and cingulum bundle and medial frontal to the subcortical region. Also using patient-specific and connectivity-based surgical targeting, we find the optimal target location before surgery, and their response rate at 6 months improved 41% to the 72%. So final conclusion is that we could improve DBS outcome over standard anatomical approach using structure-based connectivity approach. So acknowledgement is our PI, Helen Mayberg, and I'm mainly dealing with the Euro-imaging data, and I especially thank you for Dr. Patricio Riva-Posa. He did all the DBS clinical thing, and thank you for your attention.